welcome to today's A-level biology session. Yesterday we looked at basic protein structure and amino acids and the peptide bond. Today we're going to look at how the structures change. So by now, most of you should realise from GCSE that proteins have a very specific structure and they're long chains of amino acids. And that's fine, but unfortunately for A-level you need to know a little bit more than that. The first structure, that chain of amino acids, is called the primary structure. Okay, there are actually 4 levels of organisation. So the primary structure, okay, is the chain of amino acids. I put AAs because I can't be bothered to write out amino acid. Okay, so you've got a long chain of amino acids. Now, the secondary structure is how they initially fold. The tertiary structure, or the third structure, is how they fold a bit more within themselves to form a specific shape, particularly the globular proteins, which we'll look at classifications later on. And the quaternary one is where you've got 2 or more polypeptide chains coming together to form a specific shape. A really good example of that is actually hemoglobin, because you've got 4 polypeptides there. But we'll look at that tomorrow. Today, we're going to think about what happens to our long chain of amino acids. Now, there are 2 key shapes that they can go into. All right. The first one is, well, how do we find out what the chain is initially? Sorry, jumping ahead of myself there. You can use machines now, and it can be done quite quickly, actually. They use different enzymes and break it up and can analyse which amino acids are there and what polypeptides and things like, or dipeptides, and how they must all fit together. And it can take a few hours, a little bit longer for the really big ones. But the first one was done by Sanger between I have to check this because I can never believe it, 1944 and 1954. It took him 10 years to work out the sequence of insulin. 10 years. And insulin is only 51 amino acids long. Okay. Its shape is maintained, for insulin in particular, its shape is maintained by what we call disulfide bridges. And there are 2 side groups. If you remember, the amino acids have side groups, R groups. So you've got your amine group, you've got your acid group, and then you've got another side group. And the side groups have sulfurin. And the key one for this is cysteine. Okay? And they can form what we call a disulfide bridge. Now, the cysteine, I will spell it for you because I can never say it right. Okay. It has, coming out of it, an SH. So if you've got 2 SHs that come close together, okay, normally attached to a carbon, that can be oxidized and reduced. Okay, let me get it round the right way to form a sulfide bond, and that's called a disulfide. Spell it right, it's with an F these days, it's not with a PH. You will still find lots of us get spelling it wrong, and it's disulfide bridge. Okay, so you've got your chain and it can come round on itself, and it's got a little sulfur bridge in it to hold it together. Okay, it's a disulfide bridge. Now, to form it, it's oil rig, and it's oxidation to form it, taking out those hydrogens, and it's reduction to break it apart. And now you can see why the acid base content, is actually quite important. Okay, because if you didn't have the hydrogens around, it wouldn't be reduced. So that tells you why acids can break down the structure of your amino acids. Lots of other reasons, okay, and we will be looking at more of those tomorrow. So we've got cysteine, we've got this disulfide bridge. That's part of it. But the longer chain ones, there are two different types. They can form either an alpha helix. I have to dry this off. Take the base off. Alpha helix, okay, and that is basically it's not dissimilar to the helix you get in DNA. It just forms a helix around itself, okay, spiral. Or you can get a beta pleated sheet, okay, and that just does this. It's a sheet of it, so you could draw it going backwards a bit. I can never do this. There we go. Drawing was never my strong point. Okay, so you've got it going backwards from itself, forming a pleat. How are they held together? I can hear you asking already. Okay, they're held together by hydrogen bonds. But what's a hydrogen bond? I can even hear people saying that. But hydrogen bond, what's a hydrogen bond? Hydrogen bond is a very weak bond involving the hydrogen atom. And again, it depends on the side groups. It also can be associated with the ends because you've got ammonia and you've got your carboxyl group, your amine group and your carboxyl group. Okay, and basically, if you've got a hydrogen and it's going to be attached to something that quite likes things. The classic one is actually water, hydrogen bonding in water. So if I draw a water molecule here, okay, most of you should know by now that the electrons here, particularly if you do chemistry, electrons here tend to be attracted to the oxygen. So that becomes slightly delta negative. Delta means small, and this becomes slightly delta positive. And that means that if you've got another oxygen here, and if you think about the structure, you're going to have a couple of electrons there. You get what we call a hydrogen bond formed. It's quite often just drawn like that with little dashes because you've got another water there. Okay, and it's one of the really important features of water, believe it or not, because this is how it sticks together initially nice. You get hydrogen bonding, and that's why it's less dense because the bonding is actually holding it very rigid. So yeah, hydrogen bonds are really, really important, and that's what holds these together. Okay, it could be a C double bond O group and an H forming a double bond. Okay, could be lots of different things. But that's the key point with this, is the fact that you've got hydrogen bonding holding those together. There is another option, and that's called collagen. I'm particularly interested in collagen because I used to study sheepskin, and that had lots of collagen in it, I can tell you. Trying to analyse sheepskin and remove collagen from it and process the amino acids is actually quite interesting in itself. Collagen is not quite an alpha helix. It actually has 3 chains. I don't know how I'm going to draw this. It has 3 chains wrapped around each other. It makes it a very, very strong and stable protein. And that's as far as collagen tends to go. It only has a secondary structure. So it has 3 chains. As I say, I'm trying to work out how I'm going to draw this. 3 chains wrapped around each other. I ought to just get a piece of wool out. And have I got a piece of wool here? No, I haven't got any. I've only got single ply. No, I can't see them anymore. If you get strands of wool or string and untwist it, you can see how it twists around. I'm not even going to try to draw it. So collagen doesn't go any further with its structure, but it's a very fibrous protein, and it gives your skin structure and things like that. It helps hold everything together, really. It's one of the most important proteins. Anyway, that'll do for today. You've learnt now about your alpha helix and your beta pleated sheet. That's the secondary structure of proteins. Tomorrow we're going to do the tertiary and quaternary structures, which is a lot of work. Then moving on, we'll do classification of enzymes, okay, of proteins. thanks for watching today. Hope to see you tomorrow, and yeah, have a great day in the pouring rain. If it's raining with you, it's absolutely horrible here. Yeah, as I say, we'll see you tomorrow. Thanks for watching. Like, subscribe, etc. And if you've got any questions, drop me a line. Thank you very much. Bye.